Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Nanotemper Technologies, I'd like to welcome you to Characterizing Binding Interaction between PD-1, PD-L1 directly from cell lysates using MicroScale Thermophoresis. My name is Elizabeth Lamb, and I'm the host and the moderator for today's event. Now it's my great pleasure to introduce our presenter for today. He is Dr. Romain Magnez, engineer, Onco Lille at the University of Lille. Welcome, Dr. Magnez. The presenter ball is yours.
Thank you, Elizabeth. Good morning or good afternoon, everyone. Depends on where you're from. I want to thank Nanotemper and CHI for giving me the opportunity to talk about the work we are performing in Lille. So I'm going to talk to you within the next minutes about the PD-1, PD-L1 pathway and how we use the MST to study those proteins. So the short-term recurrence of cancer may be explained by the phenomenon of resistance to treatment of the primary tumor. In the opposite, the recurrence of metastasis in a patient after a full remission phase can only be linked to the ability of tumor cells to persist after treatment. At the same time, those cells remain inactive for a very long period. So in this case, the patient may seem to have recovered, but continues to carry a very small quantity of residual tumor cells in a state of dormancy. So this persistence of dormant residual cells occurs frequently and is very difficult to detect. So succeeding in counteracting and combating this phenomenon, we enable to a remarkable advance for improving the survival of patients in the long term. So the mechanism enabling the cells to enter or leave the state of dormancy are not well understood. There is a development of a balance between proliferation and apoptosis of the cell influenced by the tumor microenvironment. So the very nature of the residual cells is also a subject of debate. These dormant cells are by definition in small quantities and they are very difficult to detect and to study. So we are facing dormant residual cells that are persistent and develop immune escape mechanism, which leads to the overexpression of proteins, such as our protein of interest, which is PD-L1. So this is some overall information about the two major proteins we are studying. PD-1 is a type 1 transmembrane protein composed of 288 amino acids. PD-L1 is also called B7H1 and is a type 1 transmembrane protein with 290 amino acids and was discovered in 1999 as a new member of the B6 molecule family. And it was also identified as the first known ligand for PD-1 the following year. So the tumor environment is highly immunosuppressive and locally regulates the anti-tumor response of T lymphocyte infiltrating the tumor. So generally, tumor-infiltrating T lymphocyte express PD-1 and tumor cell express PD-1 ligands, which are PD-L1 or PD-L2. When PD-L1 binds to PD-1, an inhibitory signal is transmitted into the T cell, which reduces cytokine production and suppresses T cell proliferation. So the tumor cells exploit this immune checkpoint pathway to inhibit the immune response. To overcome the cancer's ability to resist the immune responses, many strategies are developed, such as monoclonal antibodies targeting PD-1 or PD-L1, blocking the interaction between PD-1 and its ligand using an anti-PD-1 antibody, for example, enables restoration of this loss of immune function in cancer of oropharynx, for example. Thus, some patients may benefit from a more targeted therapeutic approach that blocks the interaction of PD-1 with its ligand. These observations have led to various clinical trials aiming to block the interaction between PD-1 and PD-L1 using the monoclonal antibodies anti-PD-1 or anti-PD-L1 in several types of cancer, such as melanoma, gastric cancer, or multiple myeloma. Several therapeutic approaches that target the PD-1, PD-L1 blocking pathway are currently being developed. We can think of anti-PD-1 antibodies such as nivolumab, or combined approaches targeting PD-1 and CTLA-4, such as ipilimumab and nivolumab too. Those treatments have quite a few side effects, such as an high cost production, for example, and can sometimes lead to hepatic autoimmune reactions or dermatologic and gastrointestinal issues. So we are investigating new recently developed PD-L1 inhibitors for their ability to block the PD-1 PD-L1 interaction to restore the immune response. As I said previously, various therapeutic approaches that target the blocking of our two proteins are currently being developed. Blocking those receptors on immune effector cells can help eliminate cancer, as described recently with regard to clinical trials evaluating those antibodies. And for example, we can quote CTLA-4 receptor, which was accepted as an anti-cancer treatment recently. And also nivolumab has gained FDA approval based on its significant tumor response in clinical trials for the treatment of melanoma. There are also low molecular mass inhibitors that have also been studied for their ability to modulate the PD-1 signaling pathway. These inhibitors are based on different types of molecules, such as microcyclic peptides or oxydiazyl derivatives. BMS has also recently shown that new PD-L1 inhibitors, which are under patent actually, can bind to PD-L1 and dissociate the PD-1 PD-L1 complex at stoichiometric concentrations. However, those compounds induce the dimerization of PD-L1 in the solution. So we are using MST to evaluate those compounds. MicroScale Thermophoresis is a biophysical technique that measures the strength of the interaction between two molecules by detecting variations in the fluorescent signal as a result of an higher laser-induced temperature change. So the range of the variation in the fluorescent signal correlates with the binding of a ligand to the fluorescent target. So basically, MST is based on the motion of a fluorescent molecule within a temperature gradient. So this is typically the type of curve we obtain with MST experiment. We have a proper amount fluorescent for the bound state and a proper amount fluorescent for the unbound state. We can then establish a dose response curve between the ligand concentration and the fluorescence variation and get access to a dissociation constant. So we decided to pick MST as our binding characterization technique for the advantages listed here. There is, of course, a broad application range from picomolar to molar. A very tiny sample consumption, just a few microliters. The fact that no immobilization of the protein of interest is required. I like to point out that the no purification requirement is a specific use of MST. You can either add a fluorescent tag to your protein of interest or use the intrinsic fluorescence of the target. So our first objective was to study the PD-1 PD-L1 pathway and get access to KD in native conditions, compare with data obtained so far in literature. And then perform high throughput screening with small inhibitors that I talked previously about. So MST can be performed with covalently attached fluorophore or purified fusion proteins. So we developed a MST protocol performed on total cell lysate. One of the major advantages of this protocol is that no purification of the targeted protein is required. So it is also a more native environment. So I'm not going to describe the protocols in details, but MST performed in cell lysate allows to quantify interactions purification-free. So we use CHO cells expressing our PD-1 GFP or PD-L1 GFP proteins that we previously transfected and lysed and diluted, and that allows us to measure the binding between our two proteins. So the binding interaction between PD-1 and PD-L1 have been studied by SPR and ITC over the years, but these investigations led to controversy regarding the values of the dissociation constant. Equilibrium binding assay were performed to determine the KD between murine PD-1 and PD-L1 and yielded a 4 micromolar dissociation constant. So the cross-species protein interaction between murine and human PD-1 has also been reported in a few studies. So in 2007, the equilibrium binding responses between PD-1 and immobilized PD-L1 or PD-L2 were measured by SPR, and nanomolar dissociation constant was obtained for the binding between PD-1 and PD-L1. More recently, PD-1 ligands were once again characterized using SPR-based assays, and the result gave 8 micromolar KD for the binding of human PD-1 to human PD-L1, and 29 micromolar affinity for the binding between murine PD-1 to murine PD-L1. So let's have a quick view into the assay development. So this is typical fluorescence check to determine if our system is sticking or not to the capillaries. As you can see in the right picture, we had some sticking at first with our fluorescent molecule due to an inappropriate buffer. Same thing with absorption that has to be avoided to determine an accurate KD. So if you're facing those types of event, you can either try another type of capillaries to try to optimize and reduce the sticking or absorption, or try to optimize the buffer you're using. So this is the requirements that have to be obtained to determine the dissociation constant. At first, a reduced noise for the baseline and a reduced noise for the saturation phase. And the overall amplitude of the signal has to be above 3 times the void noise level. Since we are using fluorescent fusion proteins, we are trying to reach the most fluorescent populations of cells we can get. So this will allow us for further dilution and will allow to reduce the background noise of the lysate. So we decided to perform clonal selection using flow cytometry to obtain a very strong fluorescent cell population. This is a very crucial step when working with cell lysate, in my opinion. You have to get the most fluorescent population of cells you can get. This will allow you further dilution. So this is the overall protocol. We started by doing some molecular biology to attach a fluorescent dye. In our case, we picked the green fluorescent protein with a little linker between our two proteins of interest, which can allow for some flexibility. Then we transfected into our CHOK1 cell line to express our protein of interest. As I've shown previously, a clonal selection by flow cytometry is performed to select cells overexpressing our GFP protein. You can now perform your protein extraction where you have to decide which lysis buffer will be the best for your protein of interest. You don't want to denature your protein or have a too low extraction yield using a too soft lysis buffer. So it really depends on which type of proteins you are studying. The fluorescent protein extracted is then used for MST assays. So you have to determine the optimal ligand concentration range to get access to the KD of your complaint. Basically, you want your highest point of your concentration range be 40 times the estimated KD, and then perform a twofold deletion series. You can also go for another type of deletion too. You can then perform a capillary scan and buffer testing, as I talked previously, and can determine a KD purification free using this protocol. So this approach is not entirely new, as GFP fusion proteins expressed into a cell line was described in the past in a July of Visualized experiment. But our aim was to adapt this protocol to study a pathway, the PD-1, PD-L1 pathway, that had never been studied so far by MST, and then perform high throughput screening using this protocol. So this is the fluorescent counts of PD-1 GFP protein in presence of different lysis buffer. The RIPA buffer is providing the higher amounts of fluorescent counts, which will allow the protein to be more diluted if needed. The lysis buffer 2 provides also a sufficient amount of fluorescence for a MicroScale Thermophoresis study. But as you can see, the first buffer, the lysis buffer 1, provides less than 100 fluorescent counts. So this is not appropriate for a thermophoresis assay. So this is some other buffer, some experiment buffer we investigated to perform MST experiments. Even a non-specialist can deduce this is pretty difficult to conclude binding with those type of curves. So the whole process to test several types of buffer to determine which one is the best fit for your case study is a very crucial step. You want to avoid, of course, those type of curves. So we tried another buffer that gave quite satisfying results. The noise is reduced for the basal phase compared to the other buffer I've shown before. The amplitude is then above the full noise level. So with a tiny concentration of additives, curves are well defined. So this was PD-1 GFP against PD-L1 at first, and then this is PD-L1 GFP against PD-1. So you can attach your fluorescent dye to either one of your partner. So this won't change the binding between your two proteins of interest. So this is what we submitted to scientific reports. And as you can see, we can determine 8 micromolar binding between PD-1 and PD-L1. The non-relevant control protein did not lead to a binding curve. So this is in agreement with the most recent data reported by SPR with 8 micromolar KD. We also get a question based on the fact that our system do not reach the saturation phase. We decided to keep this dose-response curve because if we perform a higher concentration range, we will have reached the maximum solubility of our ligand by far. As it was crucial for the reviewing, we decided to perform an overreach concentration range as a proof that the binding is complete when we push for higher concentration. We can observe a well-defined saturation phase, but we decided to keep the first version as we think this is the most rigorous one. So this is shown here as cross-species binding between various variations of PD-1 and PD-L1. Murine species gives around 8.5 micromolar KD. Cross-species 8.7 micromolar KD. And the most affine one is the human version of PD-1 and PD-L1 with 7.2 micromolar KD. So we have around 8 micromolar dissociation constant, no matter what type of species is under study. So here is the crystal structure of the PD-1, PD-L1 complex. Recent data reported by BMS prove that although the targeting of PD-1, PD-L1 interaction with small molecule was considered challenging due to a relatively flat interface of PD-1, PD-L1 interaction, it is not unfeasible. I'm not like a molecular docking expert, but this is just to show a very quick overview of the PD-1, PD-L1 binding. We determine a 15 amino acid sequence in green involved in the binding of PD-L1 with its ligand. The question was, what if we remove this 15 amino acid sequence? Do we keep the binding between our two proteins of interest? So this has been studied by MST2. The wild type PD-1 protein is shown here in brown, and in green, the deletion mutant with the 15 amino acids removed. The binding for PD-1 is completely lost with the mutant, which seems to prove that the 15 amino acid sequence is crucial for the interaction. So Domenech performed a case study using MST to screen compounds on a single-shot fragment method. This is a great case study that we decided to adapt with our pathway, the PD-1, PD-1 pathway, to screen compounds targeting our protein of interest. So I invite you to check their website for more information of how they did this. So this is the overall screening of the molecules with PD-L1. In green, we have the amount of fluorescence with the PD-L1 protein by ourselves. Basically, if a molecule binds to PD-L1, we are going to have a fluorescence variation from the initial fluorescent state. So this is, of course, the case for our reference, PD-1 described here in blue as multiple replicates. In purple, molecules that do not bind to PD-L1 are going to stay in the baseline. Molecules that bind to PD-L1 are going to induce a fluorescence variation. This is the case for those 5 molecules I've shown here. So we have screened something like 200 compounds so far with PD-L1, but also with 4 other non-relevant proteins. So every time the molecule binds with PD-L1 and PD-L1 only. The molecules that bind to PD-L1 are used on a secondary screening to determine a KD. And we are currently doing orthogonal validation using other traditional physical technique. And Nanotemper developed a new software for the analysis of single-shot screening experiments, which seems to be a very nice time-saving and a great software to perform screening assays. So I can't say much, sadly, about the molecules we identified and can't show yet those response curve at the moment for the molecule. But we discovered a micromolar to nanomolar binding molecule, which might represent someday some strong drug candidates in the future. So as a conclusion for the small molecule immune checkpoint inhibitors, we think that there is major advantages in terms of pharmacokinetics and drugability. So this brings additional methods for the treatments of cancer. And what we aim is to achieve, of course, a better therapeutic effect with those small inhibitors. So as a conclusion of the use of MST to study the PD-1, PD-L1 pathway, working with a cell lysate, you have to start by optimize your lysis buffer and what conditions are the best to extract your protein of interest within, as I said, the best conditions possible. So you might also have to optimize what I like to call the experiment buffer. In our case, the HPS buffer, which is a lot of often quoted in the literature, did not lead to satisfying result, whereas it can sometimes be the appropriate buffer to study some specific interaction. So in our case, the best buffer was a PBS buffer with a tiny concentration of additive. So the crucial step, in my opinion, when performing working with cell lysate and MST, is to optimize the fluorescence expression of our cells. You can work with a poorly expressed protein, but you won't be able to perform MST experiments without a high noise and a high background signal. So you won't be able to perform multiple experiments since you will use your full lysate basically to perform one experiment. So in my opinion, this is the very crucial step of the protocol. You can also think of kinetic of your system. Does it require a short incubation period? You can sometimes obtain better results with, for example, a 30-minute incubation period, for example. So to conclude, the best results we had so far were every time obtained with what I called a fresh lysate. We were performing the extraction in the morning, for example, and we performed MST right away after the extraction. So I hope that with all those little tips, you might be successful with working with cell lysate and MST. I want to thank all the financial support we obtained from the CERIC and the RSL, all the members of my team, and Nanotemper, of course, for the invitation, and especially Pierre Soul, that helped me a lot when I was starting MST, and is still helping me a lot nowadays. And you, of course, for your attention. So in case you want to contact me to discuss about MST or the PD-1, PD-L1 pathway, here is my information and the website too, and I'd be glad to answer to your questions.
Thank you very much, Dr. Magnez. Yes, we do have some questions. Our first question, do you think in comparison with antibodies currently being developed, that a molecule with affinity in the nanomolar range is enough for a protein-protein interaction?
We think that we can, of course, compete with antibodies, because antibodies have something like 10 picomolar affinity with our protein of interest. But we are trying to develop new molecules to go below the nanomolar affinity. So we're trying to reach the picomolar affinity with the small molecule pathway. So we won't be able to compete with, but we will be able to reach almost, we hope, the same affinity as the antibodies. We hope to less side effect.
Thank you. Our next question, do you plan on screening chemical libraries on PD-1 and PD-L1 using MST?
So we thought about that, but basically the whole process is to develop new molecules based on the structure of the molecule we previously screened. So we are not going to screen a new library, but we are going to develop with relation structure activity new molecule to make the affinity of our molecule be better. So this is not the point to screen a new library now, but to perform a new synthesis and a new drug design for the molecules we are trying to investigate.
All right. You mentioned in your presentation that you had used CHO cells for expression.
Yes.
What made you choose those particular cells?
Those cells are very nice and very easy to culture. So we thought this was the best cell line to use because yes, they are very easy to monitor and very easy to culture. So this was the appropriate cell line to use for.
Excellent. Our next question, how long does an MST experiment take, and how many molecules or ligands can you test per experiment?
So basically, it depends on if you want to do a replicate of of your test, but you can get access to a KD within something like 20 minutes. And for the screening, I think you could screen a molecule per minute if you are setting up the right assay.
Next, micromolar affinity between PD-1 and PD-L1 is between their monomer. Do you expect lysate contains some residual membrane structure which would result in some avidity effect?
Yeah, we could expect that, but we did not check yet. So yeah, maybe some residue could act on the membrane structure. So this is something that we will have to look for.
Is it always possible to use lysate preparations in MST assays?
You have to express your protein of interest into the cell lysate. So you can't use every time a cell lysate, but you have to overexpress your protein of interest. So the protein has to be fluorescent, of course, but you can also have a his-tag protein expressed into your cell lysate and use an nanotemper his-tag protein labeling kit to tag your protein with a fluorescent dye and then perform an MST experiment.
All right. Is it possible to use intrinsic TRP fluorescence?
Yes, it is possible to use the intrinsic fluorescence of your molecules. You don't have to attach obligatory fluorescent dye. You can use intrinsic fluorescence of tryptophan, for example.
Ah, perfect. All right. Well, I'd like to thank you again for your excellent presentation, Dr. Romain Magnez. I'd like to thank the folks at Nanotemper for sponsoring today's event. And most of all, I'd like to thank those of you who came and spent this time with us. Hopefully you got some answers that will make your research a bit easier going forward. So thank you again so much on behalf of Cambridge Health Tech Institute and Nanotemper. Have a great day.